Now we're going to talk about the features of a bacterial cell, and we're going to start on the outside and work our way inward. And one of the external structures that we can find on about half of the bacteria that have been identified so far is that of a flagella. So flagella are typically used for movement. They're a filament-like structure that's built from a protein that's called flagellin. And they are basically kind of like a rotary propeller that spins. And it's made up of a rod, and there are what are called ring-like structures or basal bodies that are basically embedded in the cell wall. So it's a filament-like structure that's kind of anchored in the cell wall and then extends outward from the cell, and the bacteria use it to move around, kind of propel themselves within their environment. So the length and the arrangement of the flagella can vary by species. Cells can have a single flagellum or they can have multiple flagella. And so the arrangement of the flagella helps in the identification of the organism. So here we have a couple of different terms, and I forgot to put it in presentation mode, so I'm just going to flip that on right now. So we have peritrichus is one of the terms that we use to describe the arrangement of the flagella. So peri, think of perimeter. We have flagella that are dispersed over the entire surface of the bacterial cell. And so they can be coming out of one pole of the cell or the other pole of the cell or off the sides of it. So peritrichus, kind of think of the perimeter. It's all around it that we see the bacteria, or that we see the flagella. And so in this image of a bacteria, you can see these little squiggly lines extending outward. That is the flagella of this particular bacteria, and we have them coming off at all different points around the body of the bacteria. Polar arrangements refer to having flagella that is at one pole or at both poles, the pole being the end of the cell. So a bacterial flagella arrangement can be described as monotrichous if there is a single flagella that is embedded in one pole of it and it's extending outward, as we see in this image. Or it can be amphitrichous, where we have flagella that are coming off at both poles or both ends of the cell. So amphitrichous, to kind of remember that, think of like amphibians, and that amphibians are creatures that live in water, but they also live on land. So they have this kind of dual nature where they can do both. They can live in water and they can live on land. And amphitrichous, we've got kind of we have flagella at both ends, right? So both land and water or both ends of the cell. Lophotrichous refers to a tuft. And so at one pole or one end, we see this tuft of flagella extending outward, where you have multiple flagella. And in this image, you can see several flagella extending off of one pole here. So this always looks like a person to me, where it's almost like you can see this as their face, and then they've got this kind of crazy hair kind of coming off the top of their head, right? Or Crazy Larry for Lophotrichus, with this crazy tuft of hair. So monotrichous, single flagella, amphitrichous, flagella at both ends, and Lophotrichus having a tuft of flagella at one pole. Now we said the flagella are used for movement. So prokaryotes can move in response to a wide variety of stimuli. And this includes chemical gradients, it could be light, it could be oxygen concentrations. So the movement in response to a chemical stimulus is called chemotaxis. If it's moving in response to light, it's called phototaxis. All right, so taxis has to refer to that movement. So we're going to focus on chemotaxis here. And it can be a positive chemotactic response or a negative chemotactic response. So positive meaning it's moving toward a favorable chemical stimulus, or it could be moving away from something that is not good for the bacterial cell, so it's going to move in the opposite direction of that chemical, wherever it is in its environment. So positive, moving towards it; negative, moving away from it. Now when bacteria move, they take this kind of run and tumble approach to moving. If you think about it, right, prokaryotes cannot see where they're going. They don't have eyes, so they can't say, okay, that looks like a good spot over there. I think I'll just go in that direction. They can't. They have to be able to sense their environment. They have to be able to sense what is around them. So typically they will perform a run in which they're moving in a very linear direction, and then they will stop. They will tumble, meaning they're going to kind of rotate around and change directions, and then they'll go off in another run in a linear direction once again, stop, tumble, and then do another run. So this is kind of what we're showing here. So a run is very straight. The flagella is rotating in one direction. To stop that forward progression, it's going to perform a tumble, and the bacteria then, or excuse me, the flagella then changes the rotation. It goes in the opposite direction, and this causes the cell to kind of tumble and change its direction, and then it can go forward in another run. Same thing here. We have another bacterial cell that is peritrichous with flagella all over the surface of it. All the flagella are kind of like streaming behind it and rotating in the same direction as it's performing its run. And then as it goes into a tumble, you see all the flagella are kind of flayed outward, right, helping to stop the cell from that forward progression, and then it can change directions. So just another view of this, kind of using lines and the circles. So the green line represents the run, and the kind of orangish reddish circles represent when a tumble occurs. So this first square is showing when there's no attractant at all, and in B we're showing a concentration gradient, where you can think of the color, that orange color, representing some type of chemical attractant. So this is showing a chemotactic response, a positive chemotactic response, in which it is moving towards the stimulant or the chemical that is in the environment. So as the orange color gets a little bit more concentrated, a little bit more darker, it definitely wants to go in that direction. So if we start out on the left, it's performing its run, it tumbles, right, stops its forward motion, spins around, and then happens to go into another run, spins around, goes into another run, and then tumbles. And so you can see overall the progression is towards the stimulus, but it may backtrack a little bit here and there, all right? But when it's performing these tumbles, it's trying to sense what is going on in the environment. Where is that stimulus? Where is that kind of chemical strongest? Is it on my left or is it on my right side? And let's start moving in that direction. So it's a little bit random because once again, it cannot see what is around it, what's in its environment. It has to sense it. And so stopping every so often and kind of double checking, am I going in the right direction? Am I going away from that stimulus? Is it the stimulus getting weaker, or is it getting stronger? So it has to kind of take stock of that every so often in order to be able to progress towards that stimulus. If it's a positive chemotaxic response, once again a negative, it would want to move away from it to the other side. There is another type of flagella. It's what we call a periplasmic flagella, or sometimes it's called axial filaments. And this type is only seen in spirochetes. So if you remember, the spirochetes were the ones that were tightly coiled. So this helps them to move in a corkscrew-like motion. So you will not see flagella extending off the body in spirochetes because the flagella are actually embedded in the cell. So the flagella are actually located within the layers of the cell membrane. And these flagella allow the spirochetes to kind of move in that distinctive corkscrew-like motion. So that's what you're seeing in C. All right, so they're showing those kind of reddish lines are the periplasmic flagella, and they exist in what's called the periplasmic space, which is within the membrane of the spirochete cells. So remember, flagella are used for movement, and they move in a run and tumble method. And the location of the flagella is of taxonomic importance.